Okay, well, we might get started. So welcome everyone to another installment of the OHBM Australian Chapters webinar series. We've got a fantastic, very interesting and topical webinar planned for you today with some great speakers. First, I'll just provide a bit of an overview on some updates and announcements. So as we are in the new year, we are aiming to hand over to the new executive committee, which will be in the middle of the year. That will coincide roughly with when we schedule, well, we aim to schedule our next webinar, at which point we'll also have an AGM. At that AGM, we will be voting on new committee members. And so now is the time to let you all know to start thinking about whether you would like to nominate to be part of the executive committee of the Australian chapter of OHBM. We have positions available for the chair, secretary, treasurer, and EMCR representatives. We will put out a formal call very soon, so please keep your eyes out on that. We will be publicising through Twitter and emailing the membership list. So at this stage, I just invite you to think about whether you yourself might like to put your name forward, or you know someone who you think might be a good candidate, and you can tap them on the shoulder and tell them to check it out. It's a great panel to be a part of, and a great opportunity to get involved in helping to try to bring the Australian community together. So please do consider it. And that will sort of coincide with the handover to the new committee, with the incoming chair, which will be Marta Garrido, incoming secretary Valentina Lorenzetti, treasurer Johanna Bayer, and EMCR members Stefan Bowman, Irina Adnatkevichute, Tian, and James Pang. So please keep that in mind. We also still are hoping to hold an in-person meeting at the end of the year. The plan is still for that to go ahead as a joint meeting with ACNS and experimental psychology. But obviously, the situation still is a little bit unclear about travel restrictions. And so I think over the next few months, there will be some announcements about whether that will go ahead or not. But we are working towards trying to make that happen. Any other questions, please email the OHBM email address. But without further ado, we will kick off today's proceedings. So today's webinar is about robustly correlating brain and behaviour. It's a very topical topic. And we have some really excellent speakers today that are doing some fascinating work in this area. So without further ado, I'll hand over to Johanna Bayer and Tian, who will be introducing today's speakers. So thanks, Ye.
Okay, thanks Alex. So okay, I'm Ye. Let me just introduce our very first speaker, Dr. Monica Rosenberg. So Dr. Rosenberg is an assistant professor of psychology and director of the Cognition, Attention and Brain Lab at the University of Chicago. So her research explores how we pay attention and how insights from attention research can help improve focus. In particular, she builds models that predict inter and intra-individual differences in attention and cognition from patterns of functional brain connectivity across development. So she will talk about building generalizable models of behavior from functional brain connectivity. I'm looking forward to your talks. Thank you, Monica.
Thank you so much. Let me go ahead and share my screen. Okay, can everyone see slides but not presenter notes?
Yeah, good, very good.
Okay, perfect. Well, thank you so much. I'm so excited to be here today and to see some familiar faces, and also to see some, meet some new folks as well. And I'm excited today to talk to you about my lab's work, building generalizable models of behavior from functional brain connectivity. So at the outset, I just want to be clear, fMRI isn't a crystal ball. We can't use functional MRI data to perfectly predict a person's cognitive abilities or their behavior. But a growing literature has identified patterns of functional brain connectivity, as well as activity patterns and signal in brain structure that generalize across individuals to predict aspects of our cognitive abilities and our behavior. So in my talk today, I'm not going to focus on methods for building these models. I think we'll hear some cutting edge approaches to doing so later in the webinar. Instead, I want to focus my talk on ways we can assess model generalizability. In other words, how do we know that our models generalize in ways that make them theoretically useful and also practically useful? Before I jump into the talk, I just want to highlight a number of recent excellent reviews in this area discussing the benefits of predictive modeling approaches and also some best practices for building predictive models. So what are some questions we can ask ourselves when assessing whether this brain-based model of behavior that we've developed is generalizable? Well, first and most commonly, we can ask, does our model generalize across people? Does it predict some behavioral outcome when applied to data from previously unseen individuals? We can also ask if models generalize across task conditions. So for example, imagine I build a model of working memory, and that model predicts with some degree of accuracy a person's working memory capacity from data observed during a working memory task. Does that model also predict working memory capacity if it's applied to resting state data? In other words, is it robust to the exact type of functional MRI data that I have? Importantly, we can also ask if models generalize across participant populations. For example, do they predict behavioral outcomes in patients and control participants, or children and adults? Related to that question is whether models generalize across data collection sites. A critical, again, test for establishing whether our models are robust to a particular scan site or scan parameters or even pre-processing approaches. Finally, we can ask whether models generalize across behavioral measures. So returning to the example of a model that predicts working memory capacity, it's important to ask, does my model only predict performance on this very specific change detection task, or does it predict other measures of working memory as well, suggesting that it's capturing this ability rather than performance on one very particular task? On the flip side of assessing model generalizability, we can assess model specificity. In other words, it's important to test whether our model, to sort of delineate the predictive boundaries of our model. Is it specific to a particular participant population, such as development rather than adulthood? Is it specific to our behavior of interest? So again, to the working memory model, is the model predicting working memory capacity specifically, or is it capturing something about cognitive abilities more generally? And finally, we can assess whether models predict differences between individuals, like differences in clinical symptoms or cognitive abilities, or whether they capture changes within individuals over time. Models could do both. They could be sensitive to both inter and intra-individual differences, but that doesn't have to be the case. So today I want to ask this series of questions with a case study, presenting some of my lab's work building functional connectivity models that predict attention. So especially in our Zoom world, attention is a really salient part of our everyday experience, right? We need to attend over long periods of time in order to absorb content from a lecture and perform at work and in school. But it may be really salient that sometimes it's challenging to focus, and some people have an easier time focusing for long periods of time, where other people find it difficult. At the same time, we can appreciate that our attention isn't always stable from one moment to the next, but we may fluctuate between periods of being engaged in what we're doing and being distracted or experiencing mind wandering. And so in the talk today, I'll present some evidence suggesting that functional connectivity-based models or connectome-based models can predict sustained attention. They reflect pharmacologically induced changes in attention, and they also capture dynamics of attention over time and across contexts. And as I go through my talk, I'll highlight those questions about how we assess model generalizability that I raised earlier. And so first, how do we define a model that predicts sustained attention? To do so, we brought 25 adult participants into the lab, and we had them perform a challenging sustained attention task for about 40 minutes in the scanner. In this task, they were shown a rapid series of images, and they were asked to press a button every time they saw a picture of a city, but to withhold their response every time they saw a mountain, which occurred about 10% of the time. Using the fMRI data collected during this task, we calculated each participant's pattern of whole brain functional connectivity by dividing the brain into 268 functionally defined nodes and calculating the correlation between all pairs of nodes time courses. And so that gives us, for each of the 25 people in our sample, a measure of their whole brain functional connectivity pattern as they're performing the attention task, and also a measure of their performance on the task, here operationalized as D prime or sensitivity, which is just participants' hit rate on those city scenes relative to their false alarm rate to mountains. And now the goal of this analysis was to use these functional connectivity patterns to predict task performance. And we did this with an approach known as connectome-based predictive modeling. It's been described in a protocol, and the code is available on GitHub if you're interested in applying it. Using leave one subject out cross-validation, we performed feature selection and trained models on 24 participants' task data and applied it to data from the held out individual to generate a predicted measure of performance for every person. And so that's what I'm showing you here. So on the x-axis is a person's actual performance. So higher D prime scores represent better task performance. And on the y-axis are model predictions. And as you can see, this connectome-based model explains significant variance in a person's performance on this challenging attention task. Now, this is an initial way to assess model generalizability. But what about generalizability across task states that I mentioned before? Do we actually need to give someone 40 minutes of an attention task in order to predict how well they pay attention? To ask this question, we applied models defined on 24 participants' task data to data collected at rest from the held out individual. And again, we see that the model explains significant variance in performance, suggesting that it's robust to the particular task state and telling us we don't necessarily need to give someone 40 minutes of attention task in the scanner in order to observe functional connectivity patterns that predict attentional performance. Because I'll be discussing them for the rest of my talk, I'm going to give you a brief overview of the anatomy of these network models that predicted attention. So here in orange, I'm showing the high attention network. Now this network was consistently stronger in individuals who performed better on the task across all of our cross-validation folds. In blue is the low attention network. And now this network was consistently stronger in individuals who performed poorly in the task. These network masks are available on GitHub if you're interested in applying them to your own data. So in this initial part of the study, we demonstrated that this connectivity-based model of attention generalizes across individuals from this relatively small homogeneous data set. It also generalizes across task conditions, predicting performance from task and from resting state data. Now, a critical question is whether our model is actually capturing sustained attention function in general in larger and more heterogeneous samples, or whether we've simply overfit our model to this, as I mentioned, relatively small sample. To ask this question, we turned to data from a completely different participant population. And this was a data set of children between the ages of 8 and 16. These data were publicly available as part of the ADHD 200 project, and they were collected at Peking University in Beijing. So for each child and adolescent in our sample, of which there were 113, we had a resting state functional connectivity pattern and a clinician-rated ADHD symptom score. We applied our model completely unchanged from the initial data set, and what we found was the expected negative correlation. In other words, children who experienced more frequent or more severe ADHD symptoms were actually predicted to be worse at the task if they were hypothetically to be given it. Now, this is some evidence that our model defined in this data set of adults performing a lab-based task of attention is actually generalizing to capture attention function in a totally independent population measured in a completely different way. But is our model specific to attention? Is it just predicting something more general like compliance in the scanner or general cognitive function? To assess the specificity of the model, we related predictions to ADHD scores when controlling for fluid intelligence, and we still see significant predictions. However, when we do the reverse, so when we relate model predictions to fluid intelligence when controlling for ADHD scores, we don't see a relationship. So this tells us not only is our model generalizing to predict different measures of sustained attention, it's relatively specific in that it's capturing attention rather than fluid intelligence in general. Now, this work inspired brand new work in my lab led by a postdoc who actually asked whether the model captures attention function in a larger and more heterogeneous developmental sample. In particular, Omid Cardan looked at data from the Adolescent Brain Cognitive Development Study, which is an ongoing longitudinal study that includes fMRI data collected at 21 sites across the United States. So after exclusion for head motion and visual quality control, Omid asked, each child in this sample, to what degree do they express the high attention network relative to the low attention network? Again, this is the exact same model defined in our initial sample. And he related that signature of attention network strength to performance on 2 different tasks. So in particular, he looked at attention network strength as children were performing 0 back blocks of an n-back task. So 0 back blocks of an n-back task are sort of akin to a continuous performance task, like the cities and mountains task that I presented earlier, because children simply needed to press a button in response to frequent stimuli and a different button in response to rare targets. What he found is that children who showed stronger functional connectivity signatures of attention during 0 back task blocks actually performed better on that 0 back task. Again, providing evidence that the model has generalized to a completely independent data set that captures some variance in attention task performance. Interestingly, when he related the degree to which children express these networks during a 2 back task to their performance on this task, we didn't see a relationship. Now, we hypothesized that this could be because this 2 back task is relatively challenging and sustained attention simply isn't enough in order to achieve successful performance. And so in these 2 external validation studies, I provided evidence that this initial model of sustained attention generalizes across participant populations, so adults to children and adolescents, data collection sites, generalizing from the initial data collection site at Yale University to data collected at Peking University and at 21 sites across the United States. It also generalizes across behavioral measures, capturing different measures of attention, performance on a continuous performance task, ADHD symptom scores, and 0 back task performance. So it's not specific to one particular lab task of attention. Furthermore, we have initial evidence that the model is relatively specific. So it predicts ADHD symptom scores, for example, but not fluid intelligence. And so overall, I think this series of studies provides some initial evidence that this model, based on functional connectivity patterns, predicts individual differences in sustained attention. But that leaves open this really important question that I foreshadowed at the beginning of my talk, which is, our attention function isn't stable over time. We all experience more and less engaged attentional states. In order to ask whether the same network model that captured individual differences in sustained attention is actually sensitive to changes in attention over time, I'm going to present 2 series of studies. So the first is providing some evidence that the networks are sensitive to pharmacologically induced changes in attention. So to ask whether they are sensitive to these kinds of attention changes, we took the same high and low attention networks that I presented earlier, and we asked whether their strength was modulated by the administration of an attention-enhancing drug, methylphenidate. When we compared the degree to which individuals who had been given a single dose of methylphenidate before a resting state scan to a group of unmedicated individuals, we found that individuals given methylphenidate actually expressed the high attention network more strongly and the low attention network more weakly than unmedicated peers. In other words, people given this attention-enhancing drug actually showed functional connectivity signatures of stronger sustained attention. Importantly, this was a between subject study, and so we were really interested in asking whether we see this effect within participants given drugs that affect attentional states. To ask this question, we turned to 2 very different kinds of pharmacological agents. These are anesthetic agents. So we looked at data from 2 independent data sets, one in which participants had been given propofol, and one in which they had been given sevoflurane. And now anesthetic agents have many more effects than simply affecting attention, right? They affect our other cognitive states and even states of consciousness. But one thing that they do impact is our attentional states. When we looked at how attention network strength changed between people's awake scans and their scans after the anesthetics had been administered, we actually saw the same pattern of results in both studies and the opposite pattern of results that we had seen in the methylphenidate study. So in both cases, individuals expressed functional connectivity signatures of stronger attention. So stronger high attention networks, weaker low attention networks when they were awake than after the anesthesia had been administered. Interestingly, a research assistant in my group pre-registered a replication of this effect using data available on OpenNeuro. She recently found that the effect replicated in this sample such that during both a resting state scan and a scan in which participants were listening to an audio narrative, high attention network strength was strongest when people were awake relative to when they were under deep sedation with propofol. And again, the opposite pattern of result was true in the low attention network. So this series of 4 pharmacological studies from 4 different independent data sets provide some converging evidence that the same networks that predict individual differences in sustained attention are also sensitive to changes in attentional state, in this case, changes induced by pharmacological intervention. But as we think about the ways that our attention changes over time, it's probably not that common that our attention is changing because of drug administration. Instead, it's much more common that our attention changes simply because it fluctuates. Sometimes we're distracted, sometimes we're really motivated to attend, other times we might be mind wandering. And in this last series of studies, I want to present some evidence that the same network models actually captures changes in attention over time that just naturally happen from day to day and moment to moment. As a first way to ask this question, we looked at a pretty unique data set. And so these data are available on OpenNeuro, and they include 30 different scan sessions from the same individual collected over the course of 11 months. In each of these scan sessions, this participant performed the attention task with cities and mountains that I presented at the beginning of my talk. We applied our network model of sustained attention to data from each of these scan sessions separately, and we related it to session-specific measures of attention task performance. And so what we found was a significant correlation. So in other words, the model isn't just sensitive to a person's overall level of attention function, but instead it accurately predicted, right, when was this participant's best scan in terms of their attentional state and when was their worst scan. So these kinds of analyses that I've presented so far show evidence that the model generalized to capture attention measured by ADHD symptoms and also task performance. But what about the way that attention fluctuates in more real world contexts, right? Can the model change with changes in our attentional states that we experience in everyday life? In order to begin asking this question, my lab is characterizing how changes in functional connectivity over time actually predict changes in narrative engagement. This work is led by a PhD student in my lab, Hayoung Song. So in order to ask whether functional connectivity dynamics predict attention dynamics, Hayoung looked at data from 2 fMRI data sets. So in the first, participants listened to an audio narrated story written by our collaborator, Emily Finn. And in the second, they watched the first episode of the BBC TV show, Sherlock. Because these were existing fMRI data sets, we actually didn't have a ground truth measure of how people were paying attention in the scanner. So to measure how engaging the narratives were, Hayoung did something pretty creative. She ran a new study at the University of Chicago where she brought in behavioral participants and asked them to listen to the story or watch the TV show and simply rate using a scale bar how engaged they were in the narratives. What she found is that people's self-reported engagement was relatively synchronous in both the story and the TV show. And so if I show you group average results, I think that makes the point pretty strongly, right? Self-reported engagement was synchronous across people and also fluctuated over time. We can qualitatively assess these peaks in engagement by looking at what was happening in the stories at these peaks. So for example, in the audio narrated story, we find that people's engagement tended to be higher when the character mysteriously disappeared or when there was a twist ending in the story. And in the TV show, engagement peaked when Sherlock discovered a corpse, which was a clue in the mystery. Now, the key, sort of the next step in this analysis was asking whether dynamic predictive modeling of engagement, so changes in functional connectivity over time, could actually predict this group level narrative engagement measure. So in other words, the changes in attention network connectivity predict changes in attention over time in these naturalistic contexts. So when she trained a support vector regression model to predict dynamic changes in engagement from dynamic changes in sustained attention network strength, Hyun actually found that the model significantly predicted changing engagement in the Sherlock sample. So when people were watching visual images and also listening to sound, but it didn't predict changing engagement in the audio narrated story sample when there was no visual input. So interestingly, this could suggest a boundary on the model's predictive power, right? That it's capturing visual sustained attention, but not auditory sustained attention. But because of many differences between these data sets, we can't draw that conclusion from these data alone, and we're following up on that question now. And so overall, these studies suggest that the model predicts changes in attention that just happen naturally from one day to the next, or one moment to the next as we're watching a show or listening to a story. And so just to wrap up what I've talked about today, I'm going to revisit some of the questions that I mentioned at the beginning of my talk. So again, emphasizing that when we're assessing model generalizability, it's critical to systematically assess how far they generalize in terms of generalizing across people, populations, and scan sites, and also the degree to which they're specific. And finally, assessing the degree to which they capture differences both between and within individuals. Ways that we can ask these questions include internal validation, like k-fold cross-validation, and external validation, or applying models to data from completely independent data sets. Combining insights from targeted single lab studies, like the pharmacological intervention studies I've talked about, with large open samples, like the adolescent brain cognitive development study, and dense phenotyping samples with many observations for few individuals. And finally, it's critical to not only share our data and our code, but also to share our models, including our features and feature weights, so other groups can validate the models that we generate. And so just to wrap up, I've argued today that functional brain connectivity predicts sustained attention across individuals, data sets, participant populations, and contexts, and that the same brain networks predict differences in attention both between and within individuals. And finally, I'll just advocate that you assess your own model generalizability and specificity by applying it to data from completely independent data sets and by sharing your models with others. And so with that, I'd just like to thank my collaborators and my lab at the University of Chicago, and thank you so much for your attention.
Thank you so much, Monica. This is wonderful. I do have several questions in mind, but I can say we got lots of interest from the audience, so I just read out their question first, see if I have enough time. Okay, so we got the first question from Sampras. So he asked, were sleep and alertness inside scanner considered while developing the generative model to predict sustained attention, considering that these variables have shown to impact task engagement in fMRI paradigms? What do you think?
Yes, that's a great question. So when we have task data, we can rule out, people just sleepy or failing to respond at all by asking whether our model captures attentional performance in people who are continuously responding to the task. During rest, however, actually in none of the data sets that I presented, we had eye tracking data. So we couldn't validate that people were awake. In new data that we're collecting at UChicago, we do collect eye tracking data in order to assess this. I think it's an interesting open question, the degree to which what we're doing in the scanner is influencing these trait-like behavioral predictors. So, if you look at my resting state data, would you predict that I am a relatively, I don't pay attention very well because I'm sleeping, or because even if I was sleeping, my connectivity patterns would contain a signature of my attention function? And so I think it's interesting to, moving forward, be able to quantify this and investigate it more specifically.
Good. I hope Sampras has satisfied with this answer, which is I'm satisfied. I think it's very good. I think, and I also have several questions from Mariam. I think I pronounced the name correctly. So several related questions. So first is, can you elaborate on how you define your model and elaborate on more the parameters used in the model, etc. Yeah.
Yes, that's a great question. So we define the model based on whole brain connectivity using the Shen functional atlas, which includes 268 nodes. we identified features by performing a simple correlation between edge strength and behavior across individuals in a training set. And then we simply take the average strength of the connectivity in feature sets that positively relate to behavior, negatively relate to behavior, and input those values in a linear model. It's very straightforward. Interestingly, we've compared the predictive power of this model to predictive power of models defined using many other approaches, including support vector regression, partial least squares regression. We've used other atlases. For example, Haiyang Song, the PhD student in my lab, whose work I highlighted in my talk, has tried defining the model with Yo network boundaries. And we basically find the predictive power is robust to all of these choices. And so in my talk, that's sort of why I focused on methods for assessing generalizability rather than the model definition approach itself.
Good. Thanks. And I also got a question from Joshua. So he asked whether, so if he said, if I understood correctly, the system tension model discussed was derived from a single task. Is there a role for building models based on a variety of tasks? Did you look at this? Is performance better than a model derived from a single task?
This is a great question, and it's something that we're working on now. So I wish I had a completed answer for you, but I think really the way to identify the set of connections that is necessary and sufficient for predicting behavior sort of in a generalizable way is to look at the overlap of models defined by many different labs in many different samples. And so in our lab, we're looking at the overlap between models that, for example, predict performance on the task I presented here, that predict flanker task performance, NVAC task performance, with the hope that extracting these generalizable predictive features will help us kind of zero in on the skeleton, so to speak, of the networks that best predict attention.
Great. We still have a bit of time. So I have a question, we've got a question from Alex Farnoosh. So he asked, well, quite a few questions. I read them out. So how variable are the feature weights across folds in your initial training? And the second one is, how do you generate a final set of weights? The third one is, you apply those same weights to completely independent samples in your validations, did you?
Yes, yes. Okay, so starting with the first question, how variable? They were not that variable. I wish I had the, I wish I remembered the numbers, but because they were, our initial sample was only 25 participants. So we were training on overlapping samples of 24 participants. And so in that initial sample, it wasn't that variable. We defined the model to apply to the external, every single external data set that I presented by finding features that overlapped between all of our cross-validation folds in the initial training sample, and then defining feature weights in that full sample. And then that model was always applied completely unchanged, same features, same weights to all external samples. And again, that model is available online. Did I forget a part of the question?
Yeah, I think pretty much captured. And I think we have time for the final question. So I would like to take the opportunity, if that's okay. Yes. So I saw your talk, you talk a lot about ADHD. And my background is psychiatry, so I'm very interested to know. So now the current medication, normally the methamphetamine, right, as you mentioned in your talk. So I just wondering, seems like the attention network can really, it's generalizable and very powerful, seems like. So is it possible to, if we apply some like brain modulation approach to target on those attention networks, like derived and identified from your work? Would that work? Because we know the methamphetamine has some maybe side effect, and also maybe it's a short-term effect. So is it possible for future clinical trial to?
Yes, I swear I did not plant this question, but it's such a good question. So we have an initial pilot study which came out in 2020 in Neuroimage. Dustin Shinas is the first author that actually used real-time neurofeedback to ask whether we could train people to express these connectivity signatures of attention. So people were performing the attention task, they got feedback about the degree to which they were expressing the high relative to the low attention network. And our hope was, right, you could train people to express these networks, you could improve attentional performance, and you could avoid some of the medication side effects that you mentioned. This was a very small initial pilot sample that was sort of testing the feasibility of this approach, right? Calculating over 35,000 correlations in real time. And so in that pilot study, we found that the approach sort of is technically feasible, but we didn't see an impact on behavior. But that's something I'm really interested in pursuing further.
Great, thank you so much. I think it's time I'm going to hand over to Johanna to introduce our next speaker. Thank you, Monica, again. I'll see you in the panel discussion. So we still have a lot of questions from the audience. We can discuss them further at the end of the seminar. Great, thank you.
Yeah, thank you, thank you, Monica. Now it is my great pleasure to introduce our next speaker, Dr. Nico Dosenbach. Dr. Dosenbach is an associate professor of neurology at the Washington School of Medicine. His research as a developmental systems neuroscientist is focused on pushing functional MRI, resting state functional connectivity MRI, and diffusion tensor imaging to the level of individuals. In order to create and annotate the connectomes of individuals, he's working to improve the signal to noise, spatial resolution, and replicability of fMRI, RSFC, and DTI data. And as I found out through some Googling, he's also a founding member of the so-called Midnight Scan Club. Maybe he can tell us later a bit more about that. And with this, I'm handing the mic over to Nico.
Thank you very much for the very nice intro. Let me go find my slides. It's an honor to have been invited. I was kind of hoping I could see everyone's, amazing Australian backgrounds on Zoom or something like that. But actually, there we go. All right. I hope this is coming through. This is probably the only time I'm not gonna, these are my disclosures, just research funding and equity, has nothing to do with what I'm gonna talk about today. This is what I'm gonna talk about today. It's one of the few times when Alex invited me, I think he specifically said I should talk about this. Normally it's like, well, you can talk about whatever you want. I think I know why. This is a preprint, came out on bioRxiv last August, I think. And afterwards, I'm hoping some of these people are on the audience. There's quite a commotion on Twitter. Lots of really, really good suggestions that actually got us ready for, our reviewers, and, some sort of bigger picture stuff. Like Russ essentially reviewed the paper on Twitter, or the preprint, and, drew some conclusions, and then people are worried about us causing a fMRI trashing hype, which is obviously not what we want to do. And then, of course, there was already some fMRI trashing hype. So I figured this is what I should talk about. We'll make sure this is worked by Scott Merrick, together with his dynamic dual partner, Brendan Turbo-Clements. So whenever I don't know something, they'll know it. And so when I say I mean we, and when I say we, I mean they, really. And then Damon Fair, who's a good friend of mine, whose lab did all the laborious task of pre-processing all the ABCD data, and really helped us out on the computational side. Okay, and normally I don't do it in like an outline for a 20-minute talk, but I felt like I had to do this because there's a lot of confusion about what we're talking about with this title, reproducible brain association studies, and what kind of studies we're talking about. And to make sure that people know that I'm going to get to what I'm supposed to talk about, first I'm going to talk about studies that aren't BWAS, to make sure that it's very clear that I love small n and that I love fMRI, and then get to talk about this preprint. So, I mean, I've been told that I'm saying that only extremely large and neuroimaging studies are useful. I'm not saying that. This is maybe my favorite study of all times. Brenda Milner got a Copley Prize for working on patient HMs, on patient ADEM for much of her career. It's a wild story, but it's maybe one of the strongest sort of brain behavior effects that I know of, where he was a young man who had intractable temporal epilepsy, and in the 50s, an epilepsy surgery where they removed both of his medial temporal lobes and hippocampi, and then afterwards realized that he had severe dense anterograde amnesia, and that was never done ever again. So there's an N of 1. The effect was so obvious to people, so strong, essentially perfect, that it was considered unethical to ever try this again. And now if there's medial temporal lobe resection, it's always just done unilateral, because we learned from this one case that you need at least one hippocampus to make new memories. So here's another one. This is some of our own data now. We just published this, Tim Lauman, as a patient of mine. I want people to take a look at this structural MRI and take a guess as to how this patient's doing. And just, it's pretty obvious, but the black parts inside the skull, that's all just CSF. So there's no brain there. So this patient is missing large parts of their brain bilaterally in cortex. So this is, I was talking about with HM that there can be a perfect brain behavior correlation, meaning you need at least one hippocampus to make new memories. So now we've got another striking, structural lesion. It would turn out, I don't know how there's gonna be sound on this. You don't really need the sound. A little loud, but this is the patient I just showed you the MRI for. Normally I show this video first, but he's essentially a normal kid. This is when he came to me, and really it's kind of subtle, but you can kind of see that when he's using his left hand to use the wrench, he's going fast, and when he uses his right hand, he's not going as fast. So he was referred because he was playing competitive baseball, and the coach was worried that he was just much better with his left throwing things, and thought it was some kind of elbow or nerve problem. And then we found these strokes. So in this case, pretty confusing, brain structure function or brain structure correlation, brain structure behavior correlation. But yeah, here's sort of the summary. So that didn't work here in this case, but there's also fMRI, my absolute favorite. I'll make that very clear. And fMRI is pretty amazing. I mean, I think this was one of the papers I saw when I decided to go into fMRI in the early 2000s. Randy Buckner and crew predicting subsequent memories in volunteers just based on fMRI signals and a collection of brain regions. So, in case the structure doesn't tell the full story, maybe functional MRI can really help you figure out, for example, how this patient who's missing so much of his brain can essentially be typical in his behavior. And so this is again back to patient for whom I showed the video, but this is very simple HCP style motor task. This is the right left-hand contrast. And for one, I think this is really nice looking fMRI achieved just with repeated averaging. There's no mask on this. And then you can see here that we were kind of wondering before we did this, like, where's his motor cortex? And so you can kind of see that his hyperdextrous, like his dominant left upper extremity is controlled by what's considered to be a small motor cortex on this tissue that initially was thought to potentially just be scar tissue. And then, he can still use his right side, just not as good. And that's done by motor cortex that's posteriorly displaced. So he's even been able to remap his motor cortex, but he has activations that go with him being able to do the things that, I know he can do, like play baseball or work in a machine shop. So this is like a little more formal analysis of the task fMRI, and you can see, like in blue, that's his motor cortex. It's posterior displaced into parietal cortex compared to green, where the controls are. And then on the other side, the right hemisphere, left hand, his good side, you can see that, he's lost like at least half, maybe more of the tissue normally used to control the left upper extremity, but he's doing fine. So for example, it's not about the amount of M1 you have, the location might be more important when it comes to primary motor cortex. So, and that isn't all for small n research that you can, neuroimaging research where you can also look at behavior. This is everyone's favorite. Oops, I don't know how to get to play this. Okay, I took the video out. Resting state function connectivity, which I feel like OHBM is like the official resting state function connectivity organization at this point, which doesn't even require a task paradigm. You just lay in the scanner, look at a little fixation process, something like that. Try to stay awake, try not to move, and you know, there's sort of a functional structure in the brain such that regions that are commonly coactive in behavior, they're now co-fluctuating, which gives you these correlations. And this is an example of real data. And so we did individual specific precision functional connectomics in this patient, and the results, this is our favorite infomap parcellation, but you know, we could do the Yo method too. Up top is his functional connectome, and on the bottom is an average of the controls. That's actually, I think, average from the Midnight Scan Club. So that this one mentioned the Midnight Scan Club data. And you can see that even though he's got these enormous holes in his brain, the gestalt, overall, this is actually remarkably similar. And then another thing we did, we kind of quantified how much cortical surface area is devoted to each of the canonical networks. And if you just sort of look at those kind of plots, you realize that like on a glance, he doesn't look any different, right? Which goes with his behavior, which, you know, nobody even noticed that he had an issue till he was 14. It was only because he was playing baseball. You know, if he hadn't played baseball, he wouldn't have gone to a doctor. So, you know, just one subject suggested to us that maybe this kind of relative proportioning of the networks is important, but exactly where they are, if it was an early injury with the exception of M1, so in association cortex, maybe not that important. And then one more example of low end, you know, fMRI or MRI studies that teach you things about behavior and its relation to the brain. This is a study where we did upper extremity self-casting for 2 weeks and then followed with daily scans before, during, and after. And this is just one example from one subject. We did N of 3. After the first one, it wasn't that clear, so we did 2 more, kind of like in a monkey study. And what we did is, you know, we induced essentially disconnection functionally of the left and right M1. So the middle one, middle picture, I call that the half Biswell. We made the classic resting state finding disappear, and then we brought it back and we could cut the cast off, and we had a delta R of 0.86. So that's a huge effect size. So the 3 subjects, we didn't find many people that didn't replicated 3 times. We didn't really find many people that didn't believe this, mostly just talking about what does this mean. So this is just for me to like try to strongly establish that I'm a big fan of all things fMRI. I think it can tell us a lot about behavior, and I think it just all depends on what kind of study you're doing, how big the N has to be. So now into BWAS. And the other thing we realized is the preprint didn't sort of define it as explicitly as we should have had. So we looked at the official GWAS, Genome-Wide Association Study definition. It was written up in the NIH's policy relating to GWAS. And this is what we came up with. So try to sort of specifically label or limit the types of studies that, you know, we have things to say about the required or recommended sample sizes. So studies of the associations between typical and individual variability in human brain structure or function and complex behavioral traits of psychiatric symptomatology. So first crack at it. And how did we get into all of this? It was with the ABCD study. Yeah, it's very modest. This is from their website, largest ever, almost 12,000 subjects. You know, this is obviously an enormously valuable resource for looking at BWAS. And this is sort of where it all started. This is the initial paper that Scott and Brendan had when we didn't even have the full ABCD data set pre-processed yet, but a good chunk of it. And it was really to highlight how awesomely you know, replicable the resting state functional connectivity matrices are, especially group averaged, right? So up top, there's, you know, over 1,000 subjects, it's their resting state functional connectivity. And then you do a split half, and then you look at the difference matrix all the way on the side, and it's like essentially empty. you can do the same thing for the info map parcellation at the bottom. You know, you get R's of like 0.99. So it's wild stuff. But what we noticed, and this is what Scott and Brendan noticed, is that when you then looked at the brain behavioral trait relationship, for example, here this is resting state functional activity against NIH toolbox scores. The correlation wasn't nearly as strong, which really surprised me. I hadn't like super thought about this before. And you know, there was another analysis, I think it's in this figure, that kind of showed that, you know, there's quite a bit of variability in terms of the results you get for 1,000 subjects and then another 1,000 subjects when you looked at BWAS type of things versus just looking at the fMRI data or the resting state functional activity, and that the effects, even if they replicated like first component here, weren't as big as a lot of people would have thought based on what's been out there in publications. So this kind of shook us, and we decided we had to follow up on this. Code name for this was the Manhattan Project, because I thought it was sort of going to be a little explosive or upsetting potentially. We wanted a lot of time to chase this down and work on our messaging. So here, this is now the preprint that's been out for quite a while. And what Scott and Brendan did is first they looked at univariate, brain trait associations, and just essentially describe the distribution, the histograms of the effect sizes. And you know, I think we had access to some supercomputing clusters at various universities, so they kind of did it brute force and just like ran everything you could, sort of thing. So here are the distributions, and this is just exemplars. I think in the supplement to the preprint, there's like everything you can get out of ABCD, but you know, we picked popular ones like cognitive ability. This would be, you know, NIH toolbox scores, and then psychopathology would be CBCL scores. We want to look at cortical thickness as an exemplar of a structural brain metric, and then RSFC, which is very popular as a functional brain metric. And we did, you know, we did various ways of splicing the brain, so vertex-wise, ROI-wise, network-wise. And they all overlap here. They all give you the same distribution, essentially. Second row there, you can fold this kind of information back on the brain, and you know, this sort of kind of distributed, which makes a lot of sense. And I think the thing that really got us sort of thinking about this stuff was the top row. I think these are also called vibration plots, where you do the correlation or the BWAS effect for, let's say, we started, you know, in green all the way in the corner, upper corner, cognitive ability, using cortical thickness as the brain metric, and then you go sample size from 25 to essentially the full split half, which here is around 2,000 after rigorous denoising and having to remove some subjects because of head motion and stuff like that. And you know, what you see is that for small sample sizes, the variability, if you repeatedly sample from the big sample, is quite large, and you need to go out quite a ways into the sample. Need pretty big samples for that variability to shrink down, and then at the actual effect sizes are small. Shouldn't say they would be huge for like a GWAS study, but they're smaller than I would have expected. And then we had fun with some examples, like at the bottom, for example, if you just take N of 25, you can show that if you repeatedly sample, you can get an R of 0.64, and you can get another N of 25 for the same thing. That gives you it gives you minus 0.69. So strong R, but totally opposite sign. That's all just because of sampling variability. And sampling variability is sort of, you know, I used to, you know, Scott's been into it, and Brendan, they've been into it for a long time, and I always thought it was boring. I've changed my mind. But just to show you, for example, this is just blown up one of the plots that I just showed you, one of the vibration plots. And then, you know, next to it is actually also from ABCD, it's height versus weight, right? Which are obviously strongly correlated around like 0.4 or something. But you actually need quite a bit of quite a bit of sample, quite a bit of subjects to shrink down the variability around the mean. Then Scott and Brendan also looked at the statistical errors for this univariate approach, and these are various curves for false negatives, type 2, false positive, for magnitude errors, and have two types of magnitude errors at the bottom, ones that are thresholded at 0.5, and then ones that are thresholded if you're thresholding at 0.5 point for only corrected. And I mean, these are sort of complicated, but in summary, independent of what kind of statistical testing you do. So this is meant to simulate, lots of groups around the world doing similar studies in relatively smaller samples. What's the range of findings they could have and the statistical errors that would crop up, and really, to shrink down the inflation of your effect sizes and reduce the errors. You need to get pretty far out onto the right on the x-axis. I would say surprising to me. And then another one here is sign errors. So, if you're not bound for only correcting, small samples, you can have a fairly large percentage of your results, if you're simulating studies across different groups, have even just the wrong sign. So if you then just take top 1% of univariate associations, and you do our sample replication, you can quantify. This is now using the full sample, so 2,000 for each split half, or roughly 2,000. You can then quantify like how much the univariate effects are still inflated compared to what you're going to see in your out of sample replication, and it's not a huge R effect, but given the overall effect size, it's actually inflated by almost 50% when you're using the full sample available from ABCD. So then, of course, univariate doesn't get anybody out of bed anymore. So what about multivariate approaches, which are obviously much more powerful? So Scott and Brendan also took a look at those, and did lots of variations of this. I'm just going to highlight a few. So SVR is very popular, very powerful, pretty good against overfitting. And so here what I'm showing you is now SVR using cortical thickness against cognitive abilities and nice toolbox. And on the y-axis, you've got the discovery or in-sample correlation. And on the x-axis, you've got the replication correlation, so the out-of-sample. I just want you to note that the discovery, the y-axis runs from 0.2 to 1. And then because the replication R's are shrunk down so much compared to the in-sample, we cut off the x-axis at just 0.4. And then the color shows you the sample size, so from 25 to the whole shebang. And then the red and black dots just show you markers of where you would be with a sample of 200 or then the full sample. So that was cognitive ability, the psychopathology, and the gray dotted line is P of 0.01 for permutation null. So, for example, cognitive ability using SVR against cortical thickness, you see a lot of smaller sample sizes. It doesn't cross replication, but then you get some replication with the larger sample sizes quite nicely. But they're, the out of sample R is shrunk quite a bit. For psychopathology, which is just the CBCL, which is a pan-filtered out questionnaire, not so much. It doesn't look so good. You get this up-down line. Then we also tried CCA, where for the CBCL, which is psychopathology, you get more or less the same result as SVR, but for the toolbox, it looks like you just get a lot more overfitting in the in-sample, so quite the range, to 0.9 to like 0.35 or something in sample, but then really nothing above 0.4 out of sample. But there's some stuff that replicates. Here's support is SVR for resting function connectivity, and you can see SVR, resting state function connectivity against cognitive ability, does really quite well. You know, the effects are still shrunken, but for example, when you look at the black dot, you know, to be something where you have an in sample of 0.6 and an out of sample of almost, you know, like 0.35-ish. That's respectable. Psychopathology using SVR and RSFC does start showing some statistically significant replication, but is not nearly as impressive as cognitive ability. And then here's sort of the round out the box using CCA, resting state function connectivity, and NIH toolbox. You know, you get with the higher sample sizes, CCA does pretty nice, and you get replication. Although you can see by the shape of the plot that there's overfitting on the in sample, and then CSVCL doesn't do nearly as well as the NIH toolbox with psychopathology measures. So this is sort of the, I want to be very careful with this, sort of something that I think is very empirical from looking at ABCD, where SVR did a little better than CCA when it came against protecting against overfitting, and they both did way better than univariate. Obviously, as long as you had big samples, the toolbox did well compared to the CBCL, and then resting state function connectivity did well compared to critical thickness. And then you can do the same kind of plot where you look at the effect size inflation when you do an out of sample replication. Now this is for multivariate. Now this time it's top 5%. And I don't have it as percent, but like the mean shrinkage in your correlation out of sample is 0.33. One other thing that we've been asked for, which I thought was pretty interesting, these are the feature weights for SVRs and how they replicate relative, you know, out of sample relative to sample size on the x-axis. And, you know, for cognitive ability, if you do the full sample, you know, you get into like 0.3 something correlation between the feature weights. And out of sample and psychopathology, it's not quite as great, but if you're using small sample, the feature weights don't really correlate all that well. And this is sort of the summary of effective sample size on various you know, measures that might be important. So for example, at 4,000, sample size of 4,000, you know, your correlation out of sample would be around 60% of your in-sample correlation for SVR. That's sort of the burgundy line. You know, your statistical errors, your cumulative statistical errors really start dropping off after you hit sort of 1,000. And then, you know, sampling variability is really sort of the main enemy here. And this is the lab. You know, I highlighted Scott and Brendan, but other folks obviously helped out with this too. And or should I stop sharing?
Yeah, thank you, Nico, for this great talk. I would encourage everyone who has questions to type them into the Q&A. But while we are waiting, maybe I can ask a question. So in these, like the ABCD study is a study where data was collected at multiple scanning sites. Do you think that maybe variability induced by these multiple scanning sites attenuates the effect sizes that we find? So do we exchange like variability due to small sample sizes by variability due to like between site differences?
That's a great question, and we've gotten that question before. It's a brilliant question. We were worried about it. So Scott and Brendan did some analyses. I just pulled up. There's a lot on here, but essentially, what we were able to do is we compared HCP, which is single scan of single site. It was not WashU on the same Skyra versus ABCD, and we find the exact same effect size distributions and relationships to sample size. And then the other thing we did, because the ABCD has 21 sites, but some of the sites were quite efficient and scanned a lot of kits, you can also look at single site data and extrapolate, and we found the same thing. So what I would say is that for ABCD, what this means is this is a serious concern, but they seem to have done a really good job capturing the underlying population distribution and just standardizing, harmonizing across sites, which I remember there was a lot of work that went into that, making sure everybody had the similar most standard types and the sequences were all standardized and stuff like that. And it seems to have made a difference.
Yeah, great, thank you. We have another question by Tom Johnstone. He asks, what do you think are the implications for editors and reviewers of fMRI studies? How should they use these results to assess the reliability, validity of study results from smaller studies?
Oh, that's a tough question. You know, I don't want to claim to have really answers. I mean, we've been thinking about this. I think it's going to be a group effort. I think with, you know, nobody could only know the true effect sizes once we have these enormous samples. And, you know, the ABCD is $350 million still. Nobody was going to do any of this until a funding agent like the NIH was going to pony up. So, you know, now that we know, I think everybody's already doing this, you know, whenever you can, everybody should try to get the very biggest sample for a BOS type study, or think about ways, you know, Monica talked about some of them, I talked about some of them, to boost your effect size, which would be, because this is all dependent on the effect size, right? If you have a huge effect, like bilateral temporal lobectomy, the effect is perfect. If you do that, somebody will have aneroid amnesia, and of one is enough, right? So if you like give a medication or do some kind of intervention, right away you can boost your effect size, you reduce the sample size. But if you're just doing, you know, cross-sectional population based, then it looks like you need big samples. And, you know, on the positive, I mean, genomics went through this. We sort of got collaborators who went through that sort of 2008 and onwards, and who are genomicists, and they figured it out. And, you know, they started really getting much larger samples, you know, doing more standardization, consortia, enforced data sharing, that kind of stuff. And their effects are at least 10 times smaller than ours. The good news is, which makes sense, is that imaging, especially functional imaging, is so much closer to the behavioral trait, the phenotype, than genetics, that we will never need samples nearly as big as theirs. Yeah, for the editors, that's a tough question. I think split half applications are good. I think Emily Finn on Twitter suggested like focusing more on the out of sample R, which you can really bank on much more. Is a good idea. Reporting effect sizes, not just p-values. You know, I was just copying whatever genomics did 10, 12 years ago. They seem to be doing well.
Yeah, sounds good. Yeah, thank you. I think in the interest of time, we move on. But thank you for your talk, Nico, and looking forward to meet you and see you in the panel discussion.
Thanks.
Great, thanks for all our two senior international speakers. So it's the time for our local wonderful researchers, EMCRs. So now I first introduce Sina Mansour. So Sina is a PhD candidate at the University of Melbourne. His PhD mainly focused on implementing a high-resolution network neuroscience framework to model the human connectome. His research aims to improve computational models that relate brain connectivity to individual differences, influencing behavior traits and psychopathologies. Thank you, Sina. It's your time.
Thank you very much here for your introduction. Let me just share my slides. Okay, so hello everyone. Today I will be talking about high resolution connectivity, which is a novel method proposed that can be used as a biomarker of brain behavior associations. So to give you a little bit of background, we've seen that brain connectivity mapping in general and modeling the human brain as a network has become a normal way of studying behavioral traits in humans and the associations with neuroimaging. And the main reason for that is that it was seen by lots of previous research that network characteristics can actually accurately predict behavioral differences. So just to give you a few different examples, we've seen that characteristics of some brain networks, such as modularity, global connectivity, and communication efficiency, are able to significantly predict executive functioning power, cognitive control, and general intellectual performance. Monica today has also given us another example of how sustained attention can be also predicted from functional connectivity network characteristics. But the question that mainly want to talk about today lies deep within the connectome building blocks itself. So we know that the human brain network, just like any other network, is built on edges and nodes. The edges mainly explain the associations between different brain regions, either structural or functional, and the nodes basically identify different brain regions in general. And the very common way that the nodes normally are defined in a human brain connectome is from a brain atlas, which basically divides the whole brain into regions that are expected to have segregated function and structure. And then each separate region is used as a node of the connectome that is built over it. However, we've seen that in the last couple of decades, quite a few different brain atlases are developed, and in the future, I'm sure that more atlases will also be developed, and we can see that there's a wide variation between these atlases in terms of the number of regions and how they cover the whole cortical area. And also there have been a variety of studies pointing out that the choice of atlas itself can influence the statistical findings of the study. So in other words, if a study has a finding that is a connectivity-based study, and the atlas of that study is changed, the findings might also vary after changing the atlas, and that's a little bit concerning. So what are we proposing here is a alternative to the common traditional atlas-based connectivity methods, which is an atlas-free connectome implementation. Now, what we did is we took advantage of the high-resolution cortical surface mesh of the human brain, and we initially used every vertex of that cortex as a node of a high-resolution brain connectome. So in the picture here, every single dot represents a single node of a high-resolution brain connectivity network. Now, as you can expect, the high-resolution connectome will end up being much larger, and it would be computationally harder to be processed, and that's one of the reasons that in the past this high resolution method was not used as commonly. And we have proposed some new methods based on matrix representation, sparse matrix representations that can help with working with these high resolution connectomes. So having built the tools to map high resolution connectomes, we aimed for a study to have a systematic evaluation of behavior prediction performance of atlas-based connectomes compared to this high-resolution alternative, to see if there's actually any useful information by going to this higher resolution and spending more computational power on building the connectome and analyzing it. So what we ended up doing is we used data from 1,000 different individuals of the Human Connectome project, and we mapped resting state functional connectivity and structural connectivity for all individuals in two different sets of resolutions, both atlas resolution and a high resolution. So from all of those, the two sets of connectivities for both structural and functional were produced. And as we can see, the initial resolution of these two connectivities are different. So in order to be able to have a fair comparison between the two, we used an intermediary similarity metric, which basically identified how similar the connectivity fingerprints of two different individuals were in the sample. And then we used this similarity information for a behavior prediction analysis. Now, for behavior prediction, we tried to implement an out-of-sample behavior prediction method that used a wide range of different behavioral measures to sort of give us an overview of how general behavior prediction varies between high resolution, low resolution, and also functional and structural connectivity. So we took a look at age and fluid intelligence, but also implemented a data-driven component decomposition of the human connectome behavioral measures into 5 main components of cognition: illicit substance use, tobacco use, personality, and mental health. These components in itself were independent, nearly independent, and orthogonal. Once these components were extracted, we used out-of-sample prediction to evaluate the behavioral prediction accuracy. We ran 100 random splits of train and test, and each split consisted of 300 training participants and 100 testing participants. Now, the reason we chose these two numbers was that we wanted to ensure that the subjects within train, test, and in between were not familiarly related. And then we did nested cross-validation to do parameter tuning for all our machine learning models. And in order not to be biased towards a specific machine learning model, we built a consensus for testing, which basically combined the results of predictions from lasso, elastic net regression, and SVR, and the average performance across all these different machine learning models was reported as the final prediction performance. And we mainly focus on prediction accuracy indicated by Pearson correlation of the predicted behavioral measure and the original behavioral measure observed in the sample. And so to give you a little bit of what this study findings are, first of all, let me explain what this figure is showing. So on the y-axis, we can see structural connectivity on the left and functional connectivity on the right, and on the x-axis, and on the y-axis, we see prediction accuracy. So higher values are better behavioral predictions. And on the right in pink, we see the high resolution performance, and in purple, we see the Atlas resolution performance. So having a look at this overall prediction plot, we can see that the first finding of our study was that actually going to the high resolution for both structural and functional connectivity can provide better predictions of overall behavioral associations in human brain connectivity, which suggests using high resolution connectomic methods for behavior prediction in future. And another interesting observation that we had in our sample was that behavior prediction itself was context dependent. So looking at the example here, we can see that when predicting for the component of cognition in our sample, both functional and structural connectivity, even in the Atlas resolution, we're able to provide significant predictions compared to the null. And I forgot to mention, but these dashed red lines basically indicate the null hypothesis of the null hypothesis. So anything that goes above rejects the null and is a significant prediction. But we can see that for the case of tobacco use in mental health, only one modality, and especially only one resolution, can provide significant behavioral predictions. So for tobacco use, functional connectivity was the only measure that was actually able to predict differences in behavior of tobacco use compared to null. Whereas for mental health, we see that the opposite, and that the structural connectivity is the measure that's better at predicting, and only high resolution structural connectivity can provide significant predictions at this level. So, and given that the Human Connectome project data also provided a wide range of anatomical properties, we wanted to also draw a comparison between features of connectivity compared to features of anatomy like cortical thickness, sulcal depth, cortical curvature, and myelination. And we observed that in overall behavioral predictions, structural and functional connectivity provide a far more better prediction, accuracy, and performance compared to cortical thickness and other measures of anatomical differences in the human brain, which is also somewhat in line with what Nico just presented, in that functional connectivity was better than cortical thickness in doing behavioral analyses of the human brain. So just to sum up the findings of this study, we conducted a high-resolution connectivity mapping analyses and compared the results in brain behavior associations with the traditional Atlas-based alternative. And we noticed that high-resolution connectivity can improve behavior predictions in general. We also noticed that behavior prediction capability is context dependent. So it depending on what behavioral measure you're trying to predict, you might need to select a specific brain measure. So either it could be functional connectivity, structural connectivity, or even different modalities of cortical thickness. We observed that the prediction accuracy is also context dependent. We also noticed that connectivity in general is a better predictor of behavior compared to the anatomical measures that could be used. And finally, all the tools that are built for high resolution connectivity mapping are made available online. So feel free to use them in your future studies if you plan to implement a high resolution connectivity approach. I would finally like to thank all my collaborators without whom this project wouldn't have been possible. The paper is now published and available online. Also, all the codes are available online. And if you needed any help, you could reach out to me by my email. Thank you very much for listening, and it was an honor presenting my work here.
Thank you very much, Sina. Wonderful work. I'm quite familiar with this work. So I got some questions from the audience. So I got one question from Johannes. So he asked about the how is it now defined? I think in one of your.
So let me just go back to one of the slides. Okay, so the way the null is defined. So given that we have a similarity measure, we created a random distribution of possible similarity measures. So we produced random covariance matrices, and we did permutation analyses to get a null based on those random similarities. So the similarity from an actual connectivity matrix was compared to a similarity that was generated from a random covariance matrix in general.
Good, thanks, Sina. We have another question from Sempra. So, was there one specific atlas that was used to compare with your high resolution data using the?
Yeah, that's actually a good question. And in the paper, we have used the Glasser's atlas in the results that I've presented here. But we've also shown that there are two sets of questions when we're looking at the atlas. So, does the changing of atlas impact the study findings, and is our comparison valid once we change the atlases? So we've also implemented the same analysis using the Gordon's atlas, and we've noticed that the findings regarding high resolution being better than the atlas resolution in predicting behavior, and also the observation that connectivity is better than anatomical properties, both of them are replicative across different atlases. But we have also observed, so we also looked at the uniqueness pattern in these connectivity fingerprints, and we've noticed that if we change brain atlases, those patterns might not be replicated. That's also another point that indicates that there might be, so high resolution might be better than using atlas because it's not replicative across different atlases.
Great, thanks, Sina. We got another question from Braden, but for the interest of time, if you could type the answer in the Q&A box so we can move on to our next speaker. Thanks, Sina.
Yeah, thank you, thank you, Sina. So now I have the pleasure to introduce our next speaker, Jeggan Tiego. Jeggan completed postgraduate degrees in behavioral neuroscience and psychology before working in clinical neurophysiology for several years. He commenced training in clinical neuropsychology before switching to and completing a PhD. Jeggan now works as a research fellow at the Tian Institute for Brain and Mental Health at Monash University, and his research interests are in multimodal measurement of addiction and mental health problems, where he uses statistical modeling to link between genetic neuroimaging, cognition, and symptom data. So yeah, Jeggan, the stage is yours.
Wonderful. Thank you very much for the introduction. I'll just share my slide. So actually, yeah, my talk follows on quite nicely from Nico's talk earlier. And I noticed there was also a question that was applicable to this, like asking about reliability of cognitive and behavioral phenotypes. So I'm going to briefly talk about the potential limitations of increasing sample size and alternative avenues to improving power and replicability in brain behavior associations. So I guess one of the hats that I've found myself wearing over the years, and I've done a number of different things, is project management. And a long-standing concept in project management is this idea of the three levers or triple constraint model. And it's probably a bit of a belabored analogy, but the idea is that you've got these three levers that you can pull to control project outcomes, so time, budget, and scope. And we can extend that analogy, I guess, to three major and modifiable constraints in brain behavior association studies. So sample size, phenotypic precision, and MRI precision. And then we can think about the utility of pulling on these various levers and see how it affects our the robustness of our brain behavior associations. So we could pull the first lever, has been recommended, and I think that has some benefits. However, we do know that increasing sample size is very costly. MRI is about $1,000 per hour, We can combine samples across sites and form consortia, but we know that side effects are a big problem in MRI imaging as well. Furthermore, the problem with increasing sample size is that it doesn't obviate the impact of measurement error. So this strategy can still result in downward bias of the estimate that doesn't converge on the true population effect, no matter how big the sample size, so that the estimates are inconsistent. The other problem is that it may result in inflated standard errors, which reduces efficiency and can result in inconsistent and inaccurate statistical inferences, for example, false negatives. So what happens if we pull the phenotypic precision lever instead? The first potential result is that we save money, because we're improving the effect sizes, reducing our sample size requirements. The other potential result is that by increasing our phenotypic precision, our sample size requirements may substantially decrease, which clearly means more money, but also better estimates. And I'm not even going to touch the MRI precision because I'll leave that to the experts. So measurement reliability is frequently ignored in discussions of sample size and power, and yet we know from, power curves and this recent study that sample size requirements are exponentially higher as a function of lower reliability for detecting the small effect sizes that are typical in neuroimaging studies. And we can further explore these principles in relation to the Pearson's correlation coefficient, which really forms the basis of a large number of our analyses conducted in neuroimaging research literature. So due to measurement error, we can assume that R, which is the sample realization of the population parameter rho, is always a biased estimate of the true association between variables. In most cases, the measurement error will be uncorrelated between the variables, resulting in greater dispersion in the data. So the result is attenuation bias, such that the correlation coefficient will be biased downwards, resulting in attenuated effect sizes. An additional consequence of greater dispersion in the data and less clustering around the true effect is increased sampling variability, particularly at smaller sample sizes. And relatedly, we can see from the formula, the standard errors for the correlation coefficient increase as a function of smaller sample sizes and smaller effect sizes, resulting in reduced efficiency and precision of estimation. We also end up artificially underpowered and with errors in statistical inferences because the probability value of the correlation coefficient under the null hypothesis is based on this t distribution, and it uses the effect size as the numerator and the standard error as the denominator. What's more, only sampling variability will decrease with increasing sample size. The estimates themselves remain biased and inconsistent. And finally, because the resulting sample statistic is not internally valid and fails to converge on the correct population parameter, it is less likely to be replicated. So that's actually the best case scenario because a more problematic situation arises with multiplicative error in variable models. And this is where the magnitude of the error variance is systematically related to the variable itself. So a good example of how this can arise is the case of unipolar or quasi-traits, which are generally what we encounter in measurements of psychopathology. And these constructs have no meaningful variance to the lower or adaptive end of the continuum. So an example would be a bipolar trait would be intelligence or cognitive ability. You have a meaningful low end and high end. However, something like depression, an absence of depression is not the same as happiness, high happiness. So the quasi-trait properties of psychopathology constructs are quite problematic for establishing reliable brain behaviour associations because the scales used to measure them do not provide reliable measurement at the lower end of the continuum. So this is called an information function. The horizontal axis plots the latent trait theta, in this case aggressive problems as measured by the child behaviour checklist, which is standardised to the population with a mean of 0 and standard deviation of 1. Information is a metric here that quantifies measurement precision across different values of the latent trait continuum and is plotted here as a solid line. So it can easily be converted into a traditional metric of reliability using this formula, such that the information values of 2.5 equate to what would be considered traditionally as a minimal reliability of 0.6. The standard error of the estimate is inversely related to information. And you can see that there's substantial loss of measurement precision at lower trait values, such that the majority of the variance would be noise under 1 standard deviation below the mean. This is not trivial, since these measures also tend to have substantial positive skew or even 0 inflation, such that the majority of individuals will score at the lower end of the continuum and therefore have very noisy signals, and that will attenuate our brain behaviour associations. So this is coupled with the issue of non-uniform. or heteroskedasticity, which will be larger at the lower end, leading to inconsistent inference and statistical errors. So another issue is construct inhomogeneity. So if we were to measure symptom constellations and traits with most existing measures of psychopathology, we're capturing a rainbow of different sources of variance related to dissociable constructs. And these may have very different associations with brain structure and function. And so the only way to really ensure construct and criterion validity is to pass these sources of variance into homogeneous components using hierarchical models. So having highlighted these problems, I'm going to talk briefly about item response theory as a potential solution. So it's a psychometric approach traditionally used for development, evaluation, and refinement of psychological measures. And it uses mathematical models to relate item responses to underlying latent traits. So the most common model is the two-parameter logistic model, which yields two item parameters. The first is a discrimination or slope parameter, alpha. And this is analogous to a factor loading in classical test theory. It quantifies how well an item measures the underlying latent trait. Items with steeper slopes are more discriminating between different levels of the latent trait and therefore provide more information and reliable measurement. The second is the location or severity parameter beta, analogous to an item mean in classical test theory. And this indicates where along the latent trait continuum an item provides the most information and measurement reliability. So together, these two parameters are used to yield the item information functions, showing how well each item measures the latent trait and where along the continuum it provides the most measurement precision. So the good thing about item information is that it's additive and therefore sums to yield the total information function. Items can then be selected to maximize measurement precision across the latent trait continuum, across the full spectrum, whilst removing poorly performing items that merely reduce the signal to noise ratio. So IRT scale score estimates, which are analogous to factor score estimates, can then be generated using Bayesian estimation. And these have several advantages, including maximizing between subject variance compared to raw scores, as can be seen in this scatter plot. They also have higher precision than maximum likelihood estimates and can be used, they can be scaled onto the same latent trait continuum, which facilitates comparison across samples and studies. So as a brief empirical example, we had 11 different self-report measures of schizotypy, which is a liability to schizophrenia spectrum disorders in 438 individuals from whom we also obtained resting state functional connectivity data. So my colleague Sid Chopra conducted a canonical correlation analysis on these schizotypy measures and the resting state data and found no meaningful association between the brain and behaviour canonical variates. However, we know with existing measures of schizotypy, they combine heterogeneous constructs. So I use a splitting technique, which has been used quite a bit in psychiatric genetics, to pass these scales into 9 narrower homogeneous constructs known to correspond with symptom dimensions in clinical schizophrenia. And then for each dimension, such as delusions, I ran IRT analysis to select items that optimize the precision across the latent trait continuum. I then generated IRT scale score estimates for each of these dimensions. And so you can see, so on the vertical axis is the scale score, IRT scale score estimates, and the raw scores plotted on the horizontal axis. And you can see by way of this example that the IRT scale score estimates for the delusions phenotype have much better distributional properties, and they maximize the individual difference variance compared to corresponding raw scores. And so when my colleague Sid repeated the CCA using the same 29 brain PCs and the 9 IRT scale score estimates, he obtained a robust canonical correlation. So this figure shows edges that are negatively correlated with the brain variate and behavioural variate, with a proportion of edges normalised by network size displayed in the lower triangle. So lower connectivity between the default mode and salience networks and all other networks was associated with higher negative symptoms and less hallucinations and perceptual aberrations. So broadly similar with what we know from the literature. This is really just a proof of principle at this stage. The next step is to use IRT to optimize phenotypes for large scale data sets to determine if there is appreciable benefit in accuracy, precision and power. So in summary, not accounting for measurement error can result in a number of problems regularly encountered in brain behaviour association studies, and IRT may have some utility in this space, but we still really need to empirically evaluate this. Thanks.
Thank you, Jeggan, for this really interesting talk. That was really great. If anyone has questions for Jeggan, please type them in the chat. we might have the time to take 1 or 2 questions. But as long as we are waiting, I would like to ask, like, how would you like to increase, or how would you increase phenotypic precision of a sample?
So reanalysis of the data, I think, is one. So, you know, we go back and calculate, there's a couple of different issues. So I've been looking at, for example, unipolar traits, and we know that, for example, some psychopathology traits that have unipolar maladaptivity, that you can actually define the adaptive end with normal personality. So, for example, if we're looking at depression, if you link that into neuroticism, low neuroticism indexes the lower end of the depression phenotype. So that's one way you can actually increase the reliability across the latent trait continuum. So that's one option. The other is, of course, just removing items that are not indexing that trait particularly well. So we know that in certain, and that's going to be endemic to a particular sample and population. And the other thing as well is that, as I mentioned very briefly, is IRT allows you to link samples and index them onto the same latent trait continuum, and that facilitates replicability. So, yeah,
great, thank you. All right, I think in the interest of time, we could then start the panel discussion. I would ask all the speakers to come back and the audience to ask any question that you would like to be discussed in the Q&A.
Okay, great. Everyone is back. Where's Nico? Great, nice to see you back. Okay, good. While waiting for other people's questions, I think one point I would like to ask, I think would be a good start for the panel discussion is, I noticed that the effect size issue has been mentioned multiple times during the whole session. And it is true that it's kind of issue in our, when we try to study the brain behavior relationship. Even though in Nico's talk, you mentioned about, so we say the effect size typically is quite large, like around 0.8 or something when we are studying the pathology. But in terms of when we're studying the brain behavior more general, in the general population, we notice that the effect size is normally around maybe 0.1, 0.2, or 0.5 at most, which is rare, really depending on the behavior or cognitive traits we are studying. So given this fact, some question will be like, first, main question is like, we know, I think we all kind of agree that brain drives behavior. That's how human works. And so the thing is, if we're only seeing such small correlations between the brain functional structure with the behavior traits, and then in that case, is the small effect size behavior prediction really practically meaningful? Is that possible? What are we doing wrong? What are we missing? What is the way forward and the focus of future study? I think I would like to hear the opinion from all our speakers. What do you think?
And maybe because I was the biggest downer potentially, I definitely think it's meaningful. What really cheered us all up is talking to folks in genomics. Nobody thinks that GWAS or genomics in general is useless. Really what they've managed to do is get more funding and more support for their research. And obviously genes are further from complex behavioral traits than brain activity. Honestly, I mean, the biggest problem in our way, I think Jeggan said this, is scanning is so expensive. If we had the same decrement in cost every year that genomics has, we wouldn't even have a problem. I have my own conspiracy theories, like scanning is so expensive. There's only 3 companies that build these things, you know, but it's all, you know, it's sort of engineering challenges, and like how can we get organized and cooperate? But I mean, the effects are actually, to show this to some friends who are in GWAS, and they're like, oh my God, these effects are huge. You guys are so lucky. So yeah, it's just a matter of perspective. 0.1 is humongous. You can't get that in genomics ever.
How about other speakers? That's a very good point. The engineering problem really is costly. It's like at the beginning of human genome, the sequencing is super expensive, but now it's much cheaper now. So hopefully we could one day the price of MRI or even better sequencing can be even cheaper. So much more feasible in the future. So we will understand more. How about other speakers? Do you have other opinion?
I think when considering effect sizes, it's really important to consider the goal of the analysis. So you could want to predict behavior from brain features for many reasons. One would be, so for like real world applications, for clinical utility, predicting symptoms. And another is it can inform theoretical models. For example, like if we want to ask questions about how certain cognitive processes are reflected in the brain's resting state, patterns of connectivity. And so I think it's important to set standards of effect sizes for what you consider useful based on the goal of the analysis. So if an analysis that has a primarily sort of theoretical question, it may not be as high of a bar as an analysis in which we want to provide clinically useful information. And so I think it's just interesting and important to consider that there can be many different goals for this kind of predictive modeling.
Great. So I noticed that Jeggan mentioned about the precise phenotyping could be a way forward. Could you elaborate more in terms of how would you be able to, like, it might be a way to improve the effect size if we have better?
Yeah, I mean, I think we're dealing with incredibly complex phenotypes, and so the more conglomerate your phenotype, the lower the phenotypic resolution, and the more kind of dilution you're going to get in your brain behavior associations. So I think that's incredibly important for us to go back to the behavior and go, okay, exactly what is the, what level of granularity do we need to look at? How can we kind of distill this down to, a basic unit of analysis? And the other thing as well is that, and I'm going to be in danger of kind of labeling myself a dualist here, but as much as brain does drive behavior, these complex phenotypes are an emergent property of brain environment interactions. And so and there's been a bit of talk about this, is how do we improve the ecological validity of some of these paradigms, but often a lot of these behavioral phenotypes, whether they're measured by checklists or self-report, are actually measuring this emergent property of a central nervous system interacting with its environment. So how do we capture that, with more fidelity? Great
comment. And on the note of the cost of MRI scanning, we got a comment from our audience, Semprance. So he asked, what do you think about cross-modality approaches and cross-modality informed inferences, which would thereby enable cheaper modalities to be used with bigger sample sizes?
I think that's a good idea. There's a preprint that Thomas Yeo has, collapsing across resting state and task and treating task as rest, and that gives you more subjects that still have enough data to be analyzed. It helps retain sample size, and it also reduces measurement error a little bit, which helps a little bit too. I don't know. I only think about MRI. Honestly, if there's something that's way cheaper than MRI that does the same thing, that would be breakthrough, because genomics, I don't know, it's like $100 now to get a genome.
Yeah, so what's the then what's the way forward? What's the focus of future study? Any advices for the junior researchers? Are we still doing this more effect size study, or just hoping in the future we have cheaper MRI? So any advices to young researchers?
Well, I'm not on the behavior side. I'm on the MRI side. So I like what Jeggan was saying, because it seems sort of much cheaper to make the behavioral measures better. And, the toolbox did so much better than the CBCL. And I mean, I'm going to reveal myself as not being psychologically trained. And I was like, well, the CBCL is like a questionnaire. So I was like, well, what would my mom say about me? I was like, I would not want my mom filling out something about my mental state. So there's probably a lot of error in there. I mean, at least the NIH toolbox is like the child actually doing a task, which is like much closer to brain. And so I feel like I'm sure CBCL is cheap because it's just a questionnaire in terms of time compared to the toolbox, but like going through all of the things we measure on the behavior side and throwing away the stuff that is just sort of not that replicable, and working on that. Because I think that's something we can work on more than better hardware for MR scanners. That's Siemens G. Philips. I think software is different, right? There's been a lot of advances on the software side for MRI, and that's much easier and cheaper for scientists.
I think another way to sort of make it less expensive to do MRI studies is to combine data from multiple studies, right? So now you can access thousands of scans from the Adolescent Brain Cognitive Development Study and pay for storage, which is not trivial, but it's not as much as collecting that data yourself. You can access data on Open Neuro, for example, and you can build models that predict across multiple data sets, even if you yourself don't have the bandwidth or funds to collect the data needed to get replicable feature weights, for example. I also think there's a new preprint from Kwangsun Yu, who's a postdoc in the Department of Psychology at Yale, that actually builds a model that predicts task connectivity patterns from rest patterns. And so the interesting idea looking ahead might be, well, like if we only collected rest data, we may be able to use that to simulate what a connectivity pattern would look like if someone were doing a task. And that might make our connectivity patterns more useful for predicting behavior, but more cost efficient, in that we don't have to have people do many, many different tasks in the scanner.
Great. Sina, do you have any opinion on that?
Yeah, I also wanted to say that I do believe that the fact that so many different openly available data sets are now out there could itself be a way to create a proxy of not actually collecting scans yourself. And that would enable cheap ways of conducting a study. And hopefully, if enough studies on those openly available data sets just prove the fact that these MRI scans can be really useful in predicting behaviors or in explaining the human behaviors in general, I think that could then be maybe something that promotes producers such as Siemens to at least provide a way of having cheaper scans enabled in future. Because I think similar things happened with genetics. Like initially they were really, really expensive, but then there were quite a few research that pointed out how genetics could be helpful and how it would be helpful to have it for the whole population. And that's what sort of triggered this reduction in collecting genetic data.
Yeah. So thank you, Sina. We also have another question from Megan that actually goes into the direction that I find quite interesting. So she says, psychometric tests are designed to differentiate individual differences, so it depends on the fMRI aim. If it's group level, then scales might be adding noise rather than being useful. Group versus individual difference issues. Can you comment on the individual differences in fMRI? That's something I also find interesting, because I think like we had like these two approaches, that we had either like individuals that had like very specific features, like for example lesions, or we had these averages across like 2,000 individuals. But like what is actually, can we also look like at the individual, like the heterogeneity within individuals of large samples or like large data sets?
Yeah, I mean, there's quite a bit of individual variability. To the problem with resting state, which does seem to pin behavior the best, seems like, or let's just say the task or the resting state is very popular because it's easier to set up. To really describe an individual well, because of the poor SNR, you need a lot of data at the moment, which is again another engineering problem. We'll get better at that. What was a little disappointing to me is that we love collecting a lot of data. I go in a scan a lot. It's like 40 hours, 50 hours, 60 hours per person. You can't do that in a lot of people. But it does seem, I think, probably because what Jeggan was saying, that these phenotypes are so complex that that you probably with bold, which is pretty crude, you eventually bump up at what I've been calling the biological ceiling, right? So while I like collecting maybe 100 hours in a person, for certain things that's useful, like maybe it's a patient with a disorder that you only have N of 1 that you can ever find. For this kind of stuff, I think that's probably not the best use of the money. And I don't know, I haven't seen anybody that knows the sweet spot. Like if you have, if you're like the head of NIMH, and you have $350 million to spend, like how many subjects versus how much data per subject? I think that's an open question, right? Because obviously, if you stop, building aircraft carriers, we could just, scan 100,000 people's 10 hours each. That's not that expensive, but that's not going to happen. So, I think Kendrick has a nice preprint out where he talks about this, like sort of the curves of like how much total data and how do you distribute it across people. Now, probably very much depends on what you're interested in, but since nowadays people are downloading stuff, right, different kind of interests could be served by the same data set. So what's like the best all-around design? You know, how many subjects or how much data per subject? I don't know, but I think that's really important to know or guess at. I'm actually curious what people's opinions are, you know. There's been some Twitter polls.
Yeah, I think it does really much, you know, depend on what you're focused in. I mean, I'm very much in psychopathology and psychiatric neuroimaging. It's all about the individual differences, you know, and we know, for example, that most, you know, dimensions of psychopathology are in fact continuously distributed, you know, so they're not taxa. And, you know, doing group comparisons, you're really just imposing an artificial categorical distinction in that continuum. So you're just going to lose power doing that. So, and, you know, some of the people in our lab, for example, are looking at individual differences, you know, variability from, you know, group level effects across different disorders and finding that that's where, you know, that's where the meat is, you know, and we know that, for example, in psychiatric disorders, the areas that are affected tend to be the areas where you see most levels of, you know, individual difference, you know, in cortical structure and function. So maybe I'm a bit biased in that. There might be a few different disorders like neurological or, you know, disorders of Mendelian inheritance where you want to do case control analyses. But to me, you know, individual differences yields the most information.
Yeah.
Yeah, I want to argue, I mean, I think individual differences are interesting and important, and I also want to argue for intra-individual differences. Like in some cases, we can think of predicting changes over time as actually more useful than predicting changes across people if one of our goals is to identify a treatment target. So like if we find some pattern of brain connectivity that perfectly predicts an outcome, but it's not malleable in any way, then it's not useful as a potential intervention target. So combining these individual differences studies with intra-individual differences studies tracking changes in attention, like as one example over time, changes in symptomatology, you know, changes in cognitive abilities across development, I think is really critical for identifying treatment targets as well.
Yeah,
I completely agree. I mean, we've been obsessed with intra-individual, and the thing we've been trying to add is not even just wait for the natural variation. I mean, that was like Wes Podolak's micronectome study, and when we analyzed those data, we'd all expected like more variability. You know, his brain was pretty stable, and so it's like introducing interventions or some sort of perturbation. I mean, the psych patients, if it's episodic, I know a bunch of people are interested in this. You just sort of catch the ups and the downs, or medications, or behavioral therapies of some sort. That just gives you a ton of power because it's the same person. So you squeeze out all the nuisance variants, and that's much cheaper. And I think for people who are like, well, I'm not on that latest $500 million consortium, what should I do? It's much more micro. You don't need nearly as much money. It's also much lazier because you don't have to be on these conference calls with 600 people that are arguing about money. You just do your own thing, kind of, and maybe some friends. So I think that's that's definitely a way forward too.
Great. Okay, so very great discussion. And so we have been talking about more general, the feature effect size, how to improve the research field itself. So now we got a very interesting comment from Andrew Zaleski, which more focus on the model itself. So it's more, I saw he said, fantastic presentations, much focus on reproducibility of model prediction accuracy. So is the reproducibility of feature weights important and overlooked in our field? And how are we dealing with this currently? Would anyone give some opinions on that?
I think that's a great question. I had a bet with Brendan about what the out of sample correlation with the in sample of the feature weights was going to be on SVR, and I was really pessimistic. I thought it was going to be like 0.05 or something, because I just thought you could get a prediction, but you just use different information each time. So for Toolbox, it gets to like 0.35 or something. That's not terrible. Now, it's not, still quite variable. And I think what the problem is here is like interpretability. I mean, talking to sort of more old school neuroscientists, they get really annoyed at these SVR feature weights because they're like, what's it mean? What are the units? You know, so that's why, that's why they're like, well, just give me one part of the brain, because I kind of know what it's supposed to do based on animal work, and I can wrap my head around it and I can explain it. So this idea that, like, from an engineering and medical standpoint, if you can predict things, that's awesome. But I think some people who aren't as into that, they're just like, well, what's it mean? How much does it help us to interpret the brain? And I think there, the reproducibility, the weightings is really important.
I totally agree. And I think that it's true that the accuracy of your model doesn't tell you necessarily anything about how reproducible your weights are. And you could get good predictive power based on totally different feature patterns in different data sets. And I think that's why sharing models and looking at overlap between models trained in different samples, different data sets, can help you identify like what's replicable across different people, task types, etc.
I also wanted to say that, given that there could be samples of large sample size, one of the things that should be a necessary in future if we want to have reproducible data is to test for reproducibility within sample. So if someone is running SVR, could they replicate that in 2 subsamples of their data set? Like, are the weights similar within samples? So just what Nico presented on the correlation between the SVR weights itself, that would be useful information regarding the reproducibility of the findings.
Johanna, do you have any comments?
Yeah, so thank you. I think we have time for one more question. It's from Oren. It's a bit long. Maybe correlations with behavior are generally low because our analysis methods are still very primitive, introducing a lot of variability. Do you think that improved analysis method would lead to an improvement? There's still a long way to go in improving tractography for structural connectivity, e.g. solving the problem of kissing fibers, etc. And the same might be also true for functional connectivity, e.g. using advanced effective connectivity models, etc. What is the opinion of the panel?
I mean, yeah, absolutely. I mean, Jonathan Power has a paper out saying that multi-echo sequences can give you similar, replicability of the resting state data itself, shorter acquisition times, which would cut down cost. I think there's a lot of a lot of squeeze you can apply to the behavioral measures and the imaging measures. And then I think, eventually, just like genetics, which they're really good at, they do like 100x coverage. They don't have any errors anymore in their sequences, but they can still only explain a tiny amount of variance. There's going to be sort of, because I think what Jeggan said, the extreme complexity is going to be a biological ceiling. But, I don't know when we're going to get there, but, improving the methods will never be sad about that, I think.
Yeah, I mean, I think if I could add from, I can't talk to the MRI, optimization of algorithms and so forth, but, even from a behavioral point of view, it's been suggested that we're looking at the wrong level of analysis. So, for example, in cognition, rather than thinking about phenotypes, we should be thinking about microphenotypes. So, at a final level of granularity with cognition, we're often using really gross measures of performance, so reaction times and accuracy. Maybe we should be using computation, more computational models and that sort of thing. So drift diffusion models, that sort of thing. So that's something that we're looking into as well. So using finer grained level of analysis for the behavioral phenotypes.
I haven't tried it, but I'm theoretically a giant fan of EMAs and just measuring more and in different ways on the behavior side. I don't know, as a neurologist, like in Alzheimer's research, they eventually realized that a lot of their billion-dollar trials were failing because they were doing like a monumental status exam, 26 questions, a huge measurement variability. And so they were like, well, maybe we should measure our main outcome better after spending $500 million making a drug, so they're switching to that kind of stuff. And, I've talked to some people doing this, like Jason Heisenstadt at WashU, and seem, I don't know, much of it isn't published there, but they have like really much improved measurement of things like dementia. And that's cheap, right? It's like cell phone apps. You don't need a better MR scanner.
Any comments, Monica or Sina? Okay, great. I will still have 1 minute left. Maybe I would like to ask a very short question. So in terms, currently a lot of like prediction model were based on like high quality data, like Human Connect project with very good sequences. The thing is, a lot of like, how about can those models be applied to those legacy data that might be might acquired a few years ago, that when the technique was not great? So can we still use those? Can we still utilize those legacy data, or we have to just abandon them?
Yeah, I think that when you if you have a data set with a lot of data for a lot of subjects, that's a really good training data set for defining a model. And then you can apply that model to legacy data sets. And so even some of the data sets that I presented today were collected many years before I ever applied a model to them. And I think that, just based on the relative benefits of data quality really comes in the training sample. And once you have a model that is extracting signal relevant to some behavior, it can certainly be applied to these legacy data sets, which often offer like really unique tasks or interventions or participant populations compared to what you get in sort of large open source samples. And so I certainly think there's usefulness for looking at those data, of course, with the caveats that it's important to rule out potential confounds and confirm that you have good signal to noise and so on. But I think those are great test data sets for pre-trained models.
Thank you, Monica. Thank you very much. Any comments from other speakers, or if?
I just want to say this, thanks for organizing this. This was a lot of fun. I really enjoyed this. And, I'm like semi-obsessed with Australia, so I've never been there, but it's great to talk to Australians. Thanks, Zoom, it's like, you're right there. So I thought it was really helpful. I really enjoyed the questions.
Thank you very much.
Yeah, it was really great. Thank you all.
I think we have a bonus at the end of this webinar, so where we have some people from Massive Server. So they are going to introduce the huge facilities to facilitate the analyzing of brain imaging data. So I hand over to Johanna to introduce them.
Yeah, thank you. So like Ye said, we have one final 5-minute presentation from the Australian Characterisation Commons at Scale initiative, who will present to us, and Professor Wojtek Goscinski and Dr. Farnoosh Sadeghian will give a short overview over this newly found initiative. They will present a sub-project that aims to give Australian researchers access to large neuroimaging datasets like the Human Connectome project. And they are specifically interested to hear which datasets the OHBM community would be interested to get access to, and to hear if OHBM members would be interested in guiding this implementation.
Thanks very much for the introduction, also the opportunity to present. I'm just going to share my screen, please. Sorry, I can't. Sorry, 1 second, please. Okay, that's better. So thank you very much for having us present. This is just a really quick overview of the project, and hopefully I would hope that it's the start of a longer conversation that we could possibly have with this research community. I certainly, a lot of the discussion towards the end of today really resonated with some of the things that we're trying to do as an infrastructure project, and so I really welcome being able to present here today and just discuss what we're doing. So the project that we're running is called the Australian Characterisation Commons at Scale. It's an ARDC funded 3-year collaboration. It's a range of universities, 10 universities in fact across Australia, that have pooled some money and received some co-investment from the ARDC. And what we're doing is deploying tools on cloud and high performance computing infrastructure. We are a characterization focused project. So characterization here means really looking into things largely focused on microscopy and imaging techniques. And so obviously neuroimaging is something of particular interest, and in predecessor projects we've built quite a large community of users on facilities such as the Characterisation Virtual Laboratory and on the Massive facility and on other high-performance computing facilities as well, and we really want to continue that work. So one of the specialised programs that we're undertaking is on biomedical imaging collection work. So providing a resource for researchers of biomedical imaging collections that anybody in Australia can access, and that also provide the tools and services to process and to analyse. So this specific work package is not neuroscience specific, but it is going to do a lot of work in neuroscience. It's currently just scaling up due to COVID. Some of the parts of the program have scaled up a little bit slower, perhaps, and this is one of them. It's scaling up to 2 FTEs located at Monash in Melbourne. And its role is to provide access to biomedical imaging collections and the capabilities to process them. So what are the common imaging collections that you as a user community might need access to? What tools and services do you need access to? As part of your work, we're deploying infrastructure at 3 sites in Australia, at Massive at Monash, University of Queensland, UWA, and University of Sydney. And what training and processes perhaps does this user community, how can you benefit from training, or how can you contribute training as well? So either of those. We're also doing work on specifically helping Australian imaging data collections get built. And so there are a number of large sort of biomedical imaging collection projects that are building collections and that then both need the capacity to process them and manage them, but also want to make them public in some cases over a certain period of time, and want others to process them. And so we're helping underpin some of that work as well. So I'll just tell you about 2 of the things that immediately we can sort of maybe help with, and 2 of the things that you as a user community can also weigh in on. I'll present a little bit about the Characterisation Virtual Laboratory, and then Farnoosh will talk a little bit about the data collections work that we do. So the Characterisation Virtual Laboratory is a project that we've been running for some years, predates this project. And it's basically a virtual environment deployed over the top of high-performance computing and cloud facilities in Australia, and we're deploying it more and more broadly as time goes on. Its goal is to make HPC and cloud more accessible to a broader range of researchers. Our largest community is actually neuroscience, and so we're very happy with the fact that we've built that very large community, and it's really about this program of work to make HPC more accessible and more usable by new user communities. What we do is we develop rich online environments through remote desktop to specifically support neuroscience and other communities. We keep a very updated software stack. We provide then access under the bonnet to access specific data collections on the file system. We're integrated with a number of data management systems across Australia. So if you are collecting data, you can easily push your data from those integrated environments to this processing environment. And over time, we're deploying across multiple locations in Australia. So this very quick video just shows you, if you haven't used this before, and I know some of you, I know many of you probably will not have used this environment, but it just basically shows you a quick demo of what you're able to do. It's a web portal-based system where you can log in and access a remote desktop, which has a range of tools and processes already pre-installed for you, actively managed and so forth. And neuroimaging is a suite of tools that we work very strongly to keep up to date, and we work with a number of research groups to do that. It does provide a HPC, high performance computing scheduled environment in the back end, so you can analyze and manage your data interactively and then submit batch jobs as well. With that, I'll hand it over to Farnoosh.
Thank you, Wojtek. As Wojtek described, one of the goals of ACCS projects is to make data collections available to researchers, to neuroscience community. As part of this, we can provide access to curated data collections. At the moment, we have Human Connectome Project dataset, we have Nathan Klein Institute Rockland sample, and also we have Brain Genomics Superstruct project, which is available through the CDL at Massive. We also develop and provide technical capability and infrastructure to multi-site Australian data collections. And we are at a very early stage of doing this at the moment. We will support Australian Biomedical Imaging Research Database, which is a collaboration work with Australian Imaging Service Project. It's an initiative of the National Imaging Facility and aims to establish a database of shared imaging data for reuse. And also we will hopefully in future provide technical support or infrastructure support to Australian Epilepsy Project. And we also support institutional or closed research data collections. And these are just a few of the projects that we support at the moment, that they support HPCs and data management of the available systems available at CVL at Massive, including STARI, TRACK-FA, DaVinci, Pancreatic Cancer Biobank. Could you please go to the next slide, Wojtek? And we would really want to invite OHBM members to access these facilities that we have through the ACCS project, and we're seeking feedback from you, from all of you members of OHBM, Australian chapter of OHBM. For future collaborations, we would like to invite you to access CVL capabilities and resources. Please email us or you look at the website that is linked here, cvl.org.au. We will have the website for ACCS project very soon that can be shared with you. And by doing that, you can access to GPU and virtual desktops such as CVL and accessing to image processing software and libraries. And we can also apply for accessing to supported data collections. And we're seeking your feedback really to see if you're interested to access any biomedical imaging collections that we can make it available to you. And if you're interested to access to any tools and services that we can help you with. And if there is any training that would be useful to the OHBM community, it can be a collaboration with OHBM or we can support OHBM. And in what other broader digital infrastructure would support OHBM community? Please email us. My email is there and Wojtek's email is there. Please email us if you have any feedback, any question that you have to access to CVL sites. Thank you. Back to you, Wojtek.
Thank you.
Thank you.
Probably need to throw it back over one of our chairs.
Thank you very much. Johanna, okay, Johanna is back. All right.
Yeah. Yeah, so great. I don't know whether the audience has any questions. So now is the time to ask anything about this fantastic project. I'm really excited, actually. It sounds great. So maybe while we're waiting, I was wondering, like, it sounded a little bit like you're also looking for people to build this up, not only to service the community. Is that true? Like,
well, we do everything in partnership with the research community. So we can't build any of these environments without strong researcher feedback. And we do the projects that we're trying to run here is very much driven by researcher requirements. So there's many different levels that you could possibly interact with this. You could just be a user. You could start a conversation with us about specific projects that you're undertaking and to understand how we might be able to underpin them. And there's also a science advisory for this project specifically for this. So if you think that there might be ways that you can contribute more of your time because you have a particular interest in this, then please reach out.
Yeah, great. So we actually have two questions, one from Sampras. What is the scope to include real-time cloud-based fMRI processing as part of Massive?
Oh, so we have received that request a couple of times. We probably wouldn't do it on Massive. We'd probably do it on the Research Cloud, but it's certainly something that we've done in the past for specific projects. And if we think that there's an interest, then we could look into that.
Yep. Great. And Maria?
I think that's one that I wouldn't be able to answer in detail here because I'd need to sit down and to work it out a little bit to understand what the resourcing is and so forth. And I know that, look, there's obviously other projects funded by ARDC that are, that are aligned with what we're doing. So Australian Imaging Service is, sits right next to us. They're doing a federated XNAT deployment. And what I would encourage is we're working very closely with AIS. Let's sit down and understand maybe there's something that they're already doing. Maybe we work together on something. Maybe it's something that ACCS would do directly.
Yep. Great. And then Mariam asks, how do we get started using this platform?
Go to our website and ask for an account. It's as simple as that. The website's a bit clunky, I have to admit. We're doing a new one. It's a bit old. So be a little bit forgiving with respect to the sign-up process. It's a little clunky, but if you request an account, we'll get you up and running within a couple of days.
Awesome.
Okay.
And Vishma wants to know, how do you deal with the ethics component of this project if lots of groups are involved?
Yes. Okay, that's a really good question. Okay, so firstly, there's a lot of work that goes into the slide that Farnoosh presented with respect to the data collection. So even public data collections have a license, and there is a lot of work that we need to go through to ensure that we do due diligence. We pass on any, any IP requirements and things like that, any terms of use to users. That there is a process that we follow from a terms of use of data collections point of view. Most of that is to pass that on to the researcher and provide controlled access, right? So for you to access some data, you will need to see the terms of use and you will need to click accept in the same way that you do if you go now to the Human Connectome project and try to access their data sets. You actually need to agree to their terms of use. On the ethics side, so more the ethics side rather than the terms of use side, we want to support one project, early stage project that we want to support is the ABIRD project, which is a normative collection across NIF nodes under the NIF Human Imaging flagship. And look, that obviously has very strong ethics. We don't necessarily, us as an infrastructure project don't necessarily need to get involved in that, but we do need to be respectful of it and underpin it sometimes as well. And that comes again from terms of use. So anything that you do on our infrastructure is really by request to make sure that you see that terms of use and you are responsible ultimately for accessing that data in the same way you would be if it was on your hard drive on your laptop. Yeah,
that makes sense. Yeah, thank you. I think there are no more questions. Maybe we can hand over to Alex for a final wrap up.
Okay, can everyone hear me okay?
Yeah, very, very clear.
Okay, so well, it's been a fantastic session, really interesting discussion. I'd like to thank our international speakers, Nico and Monica, and also our homegrown speakers, Sina and Jeg, for some really fantastic talks and some considered answers to some of the questions. This is an important issue, and I think we need to keep the discussion going to improve the science that we're all doing as collectively as a field. And thanks again to Wojtek and Farnoosh for letting us know about this fantastic initiative, and hopefully the committee will start to buy in and make the most of it. So that brings us to a close. I hope you all have a fantastic day, and yeah, keep your ears peeled for a formal call for nominations for the committee and also for details of our next webinar. In the meantime, we'll see you later. And before I forget, thanks also to Johanna and Ye for doing an outstanding job on moderating this session.
Thank you very much, Alex.
Thank you.
Speakers.
Thank you.
Thank you. Bye-bye.
Bye.
Bye.
Thank you.